The boss says yes, so I start. Okay, thank you for the nice photos. Hopefully I'll see a few of them. Nice to meet you all. So the last bit, I'll show you some more stories about drug-relevant targets. I mean, there are many. Of course, there are many proteins you can talk about, but GPCRs are very important target for drugs. So I thought I will share some of my knowledge with them, with you. Although my knowledge is limited here because I never worked on GPCR, so I read papers, and this is what I'm sharing with you. Okay, so. And there's a database right here that's very useful. If you are interested in looking at it, you have it. Anyway, so GPCR, G protein coupled receptors. So first top of the receptor, top of the G protein, and then go on from there. Okay. So these are they are receptors, so they are in the membrane. So they have 7 transmembrane helices. That's the common feature of all these receptors. They are very important for signaling, because they are cross-membrane, so signal transduction is the critical function. And then 1/3 of the drugs actually target GPCRs, one or the other. There are about 800 GPCRs in humans, so even more than protein kinases, which is about 500 I just heard. So they're regulated by many different mechanisms. And another very common feature of GPCR is they are highly dynamic and flexible. And their conformational flexibility is how they function. Different molecules come in, can adjust the conformation. They have different states based on what the compound is bound to it. And that different states, different conformational states, basically reflect the different biology or biochemistry that come from the receptor, different signaling. In contrast to protein kinases, I mentioned they're all highly homologous. You can do alignment. GPCRs are much less conserved in sequence. They're conserved at the structural level, but not much conserved at the sequence level. And they, as your name suggests, G protein coupled, so these receptors coupled to G proteins, but it doesn't have to be G protein. It could be also arrestins or GERCs. Okay. So based on this database, there are 800 human GPCRs that we know of. That 860, actually more than that, 1,600 structures, 800 refined structures, whatever that means, and then not 1,000 drugs the targeted GPCR, many different disease indications. So it gives you a sense how important that is for drug discovery. So I have a new review that somebody sent me that came out this year about GPCR inhibitors or agonists, whatever you call it. And that's actually a very interesting summary of everything as well. So how do you classify GPCRs? And this is one way. Class A, B, C, D, F, O for odorant, smell, and taste, and other GPCRs. So, well, so if you look at structural information, that's what I care about. Hopefully you care as well. So there are, as I mentioned, 1,600 structures. They are mapped onto the structures of the various GPCRs. Class A, going all the way around the circle, not done yet. Another class A all the way around the circle. So class A actually is really a lot of members and a lot of structural information as well. So green means the active state has been determined. Red means the inactive state. Blue means both the active and the inactive state of GPCR have been determined. So you can sort of see that for class A, a lot of structural information. And then class B1, class B2, class C, class F, and then class T, the taste receptors, and then classless. Well, I guess you know what that means. So for the taste receptors, there are so far only 2 structures that are done recently. And then for the odorant receptors, very few are done. So that's why they're not even in here. The O, class O, is not even in here. But either way, it shows you a tremendous amount of structural information now on the GPCRs. And GPCR used to be thought of as very, very difficult to work with. First of all, they're membrane proteins. Second of all, they're highly dynamic. So if you think about getting a structure, a crystal of GPCR, you want the molecule to be the same, right? That's what I mentioned. And GPCR being so dynamic, it's hard to get them into the same shape. So it's very hard to get structures for them. So that's what you see. Up to 2011, 2012, very, very few structures are done on GPCR because it's so hard to study them. But since then, over the last 10 years or so, there's a tremendous explosion of GPCR structures. Part of that, of course, is Cryo-EM that enabled, they don't need to get crystal anymore. So to make things easier. Also, I guess now we understand how to stabilize GPCRs better. Even in the Cryo-EM, if the thing is very dynamic, you're not going to see it. So you have to have some kind of more stable conformation that you can sample on the average, get 2D classification, 3D volume, all that stuff. But either way, you have a tremendous explosion of structures on GPCRs over the past 10 years or so. And that's shown right here. Receptors alone, ligand complex, G protein, receptor bound to G proteins, and other things. Okay, so that's the GPCRs. So there are a couple of G proteins. So what are G proteins? G means GTP. Okay, so they bind guanine nucleotide, GTP or GDP, with very high affinity. It binds for dear life. It doesn't let go. It goes on, it doesn't come off. You'll see soon that's actually very important as well. So the G proteins coupled to the receptor are heterotrimeric complexes, G alpha, G beta, G gamma, 3 subunits. There are also smaller monomeric proteins, so-called GTPases, even though the GTPase activity is quite slow. But anyway, you'll see that. So for example, one of them is very, very famous, the RASP21, which I'll show you in a later slide. So just like G proteins, you have a conformational change depending on what ligand binds. Sorry, GPCRs, what I meant. Just like GPCRs, when you bind different ligands, you have different conformations. G proteins bind GDP and GTP, and there are two different conformations. So that's the signal. It's not the hydrolysis that's the signal. It's the conformation bound to the GTP or GDP that determines the conformation and the activity. So I'll show you that as well. So the GTP bound form is active, and GDP bound form is inactive. So that's how the system is controlled. So as a result, as I said, these proteins bind guanine nucleotides for dear life. They don't come off. So you need exchange factors to allow the GTP to be, so I guess imagine, well, like you can imagine on this slide. So RASP21, RAS means RAS sarcoma, so it's an oncogene that causes sarcoma. So GDP complex is inactive, and then GTP complex is active. That's how you get output. What happens is you have, as I said, this is bound for dear life. And in the cell, the concentration of GDP is much lower than GTP. So somehow, if your input signal comes in and use a guanine nucleotide exchange factor to kick out the GDP, GTP will have a better chance to get in. And then you activate the complex. And then how do you turn it off? You hydrolyze the GTP. And RAS and G protein on its own has a low GTPase activity to turn itself off. But then you can have GTPase activating proteins that turn it off faster so that the enzyme can return to the basal state. So you can imagine that this output most often is growth. So if this is stuck on, you get cancer. So the oncogenic mutations of RAS basically keep the protein in this state more. That's how it causes cancer. Get more and more growth signal, dysregulated growth signal coming out of this system. Okay? All right? So this is RAS bound to, in this case, GDP. So that's shown on the middle, in the middle here, GDP bound form inactive, and then GTP bound form is active. Basically, by going from GDP to GTP, two regions of the protein change conformation, so-called switch 1 and switch 2. They switch back and forth between the two states. This is the active state, this is the inactive state. That's all. Okay, not some enzymatic or this and that. So then how are G proteins classified? Based on their alpha subunit, which is the G protein itself. So GS means G stimulating sucking AMP production by using adenyl cyclase. GI inhibits the sucking AMP production. GQ stimulates PLC beta, phospholipase beta, and producing RP3 and diacylglycerol, other very common second messengers. And then finally, another class right here for small low GDP signaling. So then you wonder how, so I showed you this morning, how adenine is recognized by protein kinases. So G protein recognize guanine base also very, very specifically. Especially there's an NKXD motif, NKX right here, D right there. That motif is shared among many, many G proteins. You can see the NKXD is actually directly involved in binding the guanine. Especially the D, you can see right here, contact N1 and N2. So these, in the case of guanine, both are hydrogen donors, so our aspartic acid is perfect for making that bidentate hydrogen bonding interaction. So that's how you distinguish between A and G, because A cannot do this. In addition, A has an amino group here, so it's a donor. But guanine has a carbonyl oxygen, so it's a acceptor. So it talks to the main chain amide of this alanine, and also the side chain of 116, the N, the NKXD. So this side chain talks to the N7 and the O6. So that's the hydrogen bonding interaction that gives you specificity. And then you can see the base on one side of the phenylalanine stack against it. On the other side, you have a lysine side chain, the NK, the lysine, this K motif. So the hydrophobic part of it lies next to the guanine base. So hydrophobic interaction. And then the guanidinium group at the end, not guanidinium, sorry, ammonium group at the end interact with the oxygen in the ribose. So it's a very tight binding site that determines that this is a guanine binding pocket. Okay, so then that's how the guanine is recognized. And then it uses GDP or GTP, so how the phosphate on the nucleotide recognized. That's using something called a P loop, which has this conserved sequence G4 residues GK. Okay, so that interacts with the phosphate group or the nucleotide, in this case the beta phosphate or GTP or GDP. So the phosphate 100 bonds the main chain amides of this motif. There's also ionic interaction between the lysine side chain right here and the phosphate. So that's shown better here. So in the case of RAS, here the beta phosphate, here the P loop right there. So if you turn it around a little bit, you see, so G10 is the first conserved G, and then goes around. Then this is the GK glycine here, a lysine, and then there's also a serine that's quite conserved as well. You can see the 100 bonding interaction of the main chain amides. I made this figure back in 1980 something, so no color. It came from my thesis. I was a student once myself. Hopefully you understand that. Some years ago. Anyway, so you can see main chain 100 bonds to the phosphate, and then the serine and the phosphate coordinate the magnesium ion. And that's also very important. So many G proteins have a serine following the K. That serine is involved in mediating contact with the magnesium. Okay. So I said there are many, many structural GPCRs. I'll show you only two today. So this is the major breakthrough for GPCR structural biology, done from Brian Popelka. I guess you know who that is. So this is from 2011, and I think he won the Nobel Prize a few years after that. So this is you could say, a crowning achievement, because he had made great progress studying GPCR alone in the membrane. But here is the structure of GPCR bound to the G protein. So that's shown right here. Okay, so that's what we'll talk about. How does that work? So how does it work at the biochemical level? So here you have GPCR, which binds the ligand, an agonist, that causes the configuration change at the bottom that allows G protein, alpha, beta, gamma to bind. You can see it's the alpha subunit that binds. Okay. That allows the GDP to leave, and then GTP can rush in. So now you have GTP complex, now it's active, and then once GTP bound, there's a complementary change. Because of the complementary change, the G protein alpha beta gamma trimer falls apart. So alpha subunit goes talk to adenyl cyclase to make cyclic AMP, the important second messenger. And then the beta gamma subunit goes talk to cause a calcium influx, another important cell signal transduction process. And then in the end, GTP gets hydrolyzed to get GDP, and then the whole trimer forms again. It goes back to the beginning. Okay. So what they have is the structure of this state. So this 2011, so it's done by crystallography. And they did a lot of things to get the crystals. So since you know crystallography now, I'll show you what they did. So here's the protein. So GPCR up here bound to an agonist, BI167107, which is up here. Now here's G alpha, G beta, G gamma, which is in, what color is that? Purple? Okay. I was waiting for you to give an answer. Anyway, so first they use the antibody, a nanobody, to stabilize the conformation bound, so you can see that's in red. It's bound to the G beta and G gamma. So to stabilize the G protein part. Second, they introduced T4 lysozyme as part fused to the GPCR. And that helps crystal packing. Because lysozyme, as I showed you, grow beautiful crystals, likes to pack into crystals. So they took advantage of that. You can see lysozyme sits between molecules in the crystal. So without that, you're not going to get crystals either. So this is sort of their magic to use an antibody to stabilize the conformation, use lysozyme to help form crystal packing interactions. That's how they got the crystal, and that's how they got the structure. Okay? So then the question is how, so this obviously is the active state because the BI167107 is an agonist, and it bonds to the GS. So everything is in an active state. So then the question is, how does the G protein change conformation going from the inactive state to the active state? So this is the structural overlay between the active state and the inactive state. So active state in green, inactive state in blue. And if you look at, this is only the G protein part, 7 transmembrane helices. You can see TM6 has a very large complementary change going from here inactive to here active, so it opens up. And as you see later, that opening up allows the G protein to bind. Without that, they'll clash into this transmembrane helix. And then TM5, the other helix, is longer by 2 turns. So there are a lot of complementary changes by binding a different ligand to the top of the structure. So this will be extracellular, and this will be intracellular. So essentially, so the binding site typically is in the top part of the structure. As you can see from the bottom view right here, cytoplasmic view, so you can see the helix move by, in their measurement, 14 angstroms. So here is in the middle of a pocket here, and that's the helix will need to bind. So we will not spend time looking at the structure, but I have made I made some movies, so I'll just show you the movies. We'll spend the next, after this is done, after your lunch, we'll have another hour. We'll make some movies or other things. But this is to give you a sense, because that figure is too complicated, is that right? Well, maybe. So that's what I show you. So is white background okay in the back? Can you see okay? All right. So, oh, that's not great. Okay, here's the morphing. back and forth. You can see between the inactive state, which is in blue, same color scheme as they did, and green, the active state. You can see that dramatic change. You can also see putting the G alpha right here, and that's actually clashing into G alpha in the inactive state. So there is no binding site for G alpha in the inactive state, and this conformal change is essential for allowing that to bind. Okay, so I can zoom in a little bit more if you'd like. So you can see from the side view, the helix directly sits on this yellow helix, which came on the G alpha C terminus, because this is the C terminal residue, 300 something. Yeah, so that's a C terminus. So C terminal helix bind to the interface with GPCR, and that's how the alpha segment is recruited. So there are many, many interactions between this helix and the GPCR. You can also see there are changes elsewhere as well. You can see a lot of changes in the structure. Some of these may be real. Some of this may be because the resolution is not high enough, so they cannot determine the structure to high enough confidence. But this change obviously has to be real. Otherwise, there's no room for binding the ligand or binding the G alpha. Okay, so I'll leave this on because we'll come back to it. All right, so that's what I just showed you. The C terminus helix bind to the interface with GPCR. So there are many, many interactions, polar interactions, hydrophobic interactions. And if you're curious about it, I guess you can look at it as well. The structures is available on the PDB. I think I've also uploaded to your folder as well. Anyway, so that's how G alpha gets recruited. So I won't go into the detail on that. And most importantly, when you look at the structure of G alpha, it has two domains. One domain is the same size as the small GTPases. The bind GTP do the signal switch 1, switch 2. It also has the alpha helical domain. AH means alpha helical domain. Normally, on its own, the alpha helical domain folds onto the GTP molecule, so keeping it in place. So in the complex with the receptor, the whole thing swings out. So now the GTP nucleotide is no longer buried in the structure. So now it can leave. So that's sort of how the exchange would happen. So this thing swings over by about 120 degrees from being here to being there. Okay? And there are other changes as well. I guess we won't spend too much time talking about them. But the big idea is there's a binding of the ligand causes a conformational change, allowing G alpha to bind. That causes a conformational change in G alpha so that the alpha helical domain swings out, and then the guanine nucleotide can exchange. And then you may wonder, how does ligand binding causes activation or inactivation? And the inactivation is something also called the inverse agonist. It deactivates the GPCR. So you can see this is the agonist, BI compound. This is the inverse agonist. Sorry, this yellow you cannot see, pyrazolol. So overall, you can see they're quite, by looking at them side by side, you say, oh, they're very similar. But if you overlay them, focus on this one and this one. So this is bound to an inverse agonist. This is bound to an agonist. You can see the isoleucine 121 and the isoleucine 121 sits very differently. And this phenylalanine and this phenylalanine, same residue, phenylalanine 282, sits very differently. So you could say small differences, but they are certainly differences. These differences near the ligand binding site gets propagated further down, and that's how the whole system changes conformation. The system is so dynamic that many, many metastable states, if you will, and then having a compound can stabilize one or the other state. That's how you select between different states. Okay, so having structural information is also very helpful to understand how this overall process goes. Okay, so let me see whether we want to go back to that, whether I have that. Yeah, I do. So So this is the G protein part. This is the nanobody, right? And then, let me see. That's the so this is the alpha H domain here, and then the structure is on the other side. So it sort of helps you to understand the conformational changes in the enzyme or in this receptor system as a response to ligand binding. Okay, so that's the first story on G proteins. The second story, which you probably heard of, is Ozempic and Wegovy. Anyway, so that's linked to GLP-1, glucagon-like peptide 1. So it's a hormone, peptide hormone, that regulates blood sugar levels and appetite, stimulates insulin release. So that means it will reduce blood sugar levels. More importantly, it also reduces appetite and food intake. So it has really good properties. The peptide comes from pro-glucagon. It contains about 30 residues or so. But this is the sequence. This one, 31 residues. So this has all the right properties, except it's not stable. So it gets processed by DPP4, dipeptidopeptidase 4. That's what that means, DPP, dipeptidopeptidase. So it cleaves by peptide, dipeptide from the N-terminus of this peptide, and then the whole thing gets degraded. So the half-life of this is minutes. So there's no way you can make that useful as a drug because you'll be taking that every few minutes. So what did they do? Well, first, the receptor. So GLP-1 binds to GLP-1 receptor. And GLP-1 receptor agonists, such as semaglutide or Ozempic or Wegovy, have found great success in treating type 2 diabetes. And now it's also being used for obesity and other conditions. So there are a lot of positive, I mean, of course, there's important side effects as well. But so I don't know how popular it is in India here, but in the US, it's everywhere. So, and then GLP-1 receptor antagonists. So this is an agonist. Antagonists are also being studied, for example, for hyperinsulinism, the reverse, which sort of makes sense. So what did they do to GLP-1 to make it stable? So semaglutide, this is GLP-1 you saw on the previous slide. This is semaglutide or Ozempic. So they only made 3 changes to the peptide. They changed alanine to 2-amino isobutyric acid, basically alanine with 2 methyl groups on the side chain. Okay, the L and D methyl group, both are there. By having both methyl groups, DPP4 can no longer cleave here. So that's how you prevent proteolysis. And then the lysine is modified by a PEG group to enhance pharmacokinetics. And then this lysine is changed to arginine. So 3 changes. Now the half-life is 1 week in the body. So now it's a pharmaceutical relevant compound. And that's what you use. So you inject yourself with this peptide. So how does the peptide works? So here are the structures of semaglutide bound to the receptor, bound to the G protein as well. But I will not talk about G proteins anymore because we already did that. And so the semaglutide, you can see it's a very large helix, sits in the ligand binding side of the G protein. And then here you can see the modification of the lysine, some EM density for it. And here you see the, right here, the modified alanine, so 2 side chains. So that basically blocks proteolysis by DPP4. So everything else acts just like a small molecule. It binds into that side. It goes up beyond that. There's a C-terminal domain that binds some of that as well. But anyway, So that's sort of how the peptide works. It acts as a very long helix bound to the GPCR. Okay. So here you can see between the active and inactive state, there's conformational changes in the enzyme. Oh, sorry, in the GPCR. And you can see essentially here, you have the modification of the lysine. And I think I showed at the bottom. Here, modification on the alanine to make it not hydrolyzable. And the other thing I showed here in the overlay, so as you saw, GLP-1 or semaglutide is a peptide. So obviously people will also be trying to develop small molecules that bind to this receptor and cause the same effect. So in yellow here is one such small molecule, which I'll talk a little bit more later. So you bind the same area and have somewhat similar effect. So you saw large conformational changes. Actually, the conformation is very dynamic. Based on EM analysis, they can see many, many different states of the complex. That's sort of illustrated here. The extremes of the conformation of transmembrane helix 1, extracellular loop 2, how they are moving back and forth in different states of this complex. So here the GPCR in green, and then there's an extracellular domain right here, and then the semaglutide also moves back and forth. You can see going from this position to that position. So very dynamic system, even though you have a complex, still very dynamic. So here are the small molecule agonists or non-peptidic agonists of GLP-1R, a Pfizer drug, or Pfizer compound, I should say, a Chugai compound. You can see they have different properties compared to GLP-1. So they can activate something, but not other things. Well, GLP-1 can activate almost everything. But still, so small molecule will represent a different modality of treating diabetes, obesity, this and that. So how do they work? So they bind the same place right here. And this is G-beta, this is G-gamma. So G-beta, so I forgot to mention when we first talked about it. G-beta is a beta propeller, WD-40 protein. That's right here. So that's the important part of this complex with GABA to do signal transduction. Okay, so this is the Pfizer compound bound to GLP-1R, GLP-1 receptor, Cryo-EM density, and then this is where the compound binds. And more detailed interactions, polar interactions, hydrophobic interactions, this and that, and too much information, right? Moving on. So this is the comparison of all three. GLP-1R bound to the Pfizer compound, bound to the Chugai compound, and bound to GLP-1, comparing different conformations. You can see there's a lot of conformational flexibility among these different structures. So that's probably explaining why GLP-1 can do some things where these compounds cannot do the same thing, because they are causing different states of the receptor. Okay? All right. So I know I'm flying over this in superficial, but I think for you, the main idea is to give you exposure to it. If you're interested, all of these are published. You can go read the paper or download the PDF file and look at it yourself. So this gives you a sense of molecules or proteins that are important for drug discovery. So GLP-1 or semaglutide, something that came on the market over the past couple of years and has really taken at least the US by storm, and Europe as well, I think, by storm. Many people are taking them. Okay, the last story. Not GPCR anymore. We're changing field altogether. Cystic fibrosis. Have you heard of that? Yes? Okay. So it's a rare autosomal recessive genetic disease, whatever. So it impairs the clearance of mucus from lungs and other organs that leads to bacterial infections, and that's the disease, essentially. So it's caused by mutations in the CFTR gene, which is a cystic fibrosis transmembrane conductance regulator. Well, I'll just say CFTR. Most common mutation is deleting one residue, phenylalanine 508. That's in 70% of patients, that's the mutation. Here is the AlphaFold model of CFTR. You can see the color by AlphaFold scheme, blue, and then you have these orange things which I should hide. Well, first I want to sort of show the entire glory of this protein. Gigantic protein because it's almost 1,500 residues. And how do the mutations affect the function? It destabilizes the protein and also prevent trafficking of the protein from, it's a membrane protein, so it's made in the ER and trafficked to the plasma membrane. So that trafficking can also be disrupted. So essentially, even if you have protein made in the ER, it doesn't get to the membrane. That is useless. So all of these happens. And then I skip over this one here. So it's a anion transporter. It's transport the chlorodynes, other anions. And then CF or CFTR belongs to something called ABC transporters. Okay? So what are ABC transporters? ABC means ATP binding cassette. So they have a cassette or a domain that binds ATP. So this is shown, so it's actually these proteins are found from bacteria to man. So here is a bacterial system, periplasm, cytoplasm. So this transporter sits in the plasma membrane. And then there's a substrate or ligand outside the cell in the periplasm. So there's a periplasmic binding protein that recognizes the substrate, and that delivers to the receptor. And the receptor has two transmembrane domains and two NBDs, nucleotide binding domains. That's shown right here. And then each one can bind the ATP. So ATP comes in, bind, and then ATP mediates a dimerization of the NBD that causes a conformational change in the receptor. Now the ligand, the substrate can be delivered to the receptor, transporter. And then ATP hydrolysis causes a conformational change in the receptor. And now the substrate can go inside the cell. And then the whole thing cannot be recycled. Okay? This is one molecule. Even though you see two domains, everything, it's not a dimer. It's a single molecule. So that's shown right here. On the left, this is figures I made myself. So this is colored by rainbow. Okay, you know that, right? Blue N-terminus, red C-terminus. So you can see transmembrane region is blue. So N-terminus start with transmembrane region. And then this NBDs right here is green, so near the middle of the protein. And then another transmembrane domain, and then red is the second NBD. So the NBD, one from the middle, one from the end. And then the phenylalanine 508 is right here. This is part of NBD1. This residue right here, I showed right here, and sits in the large hydrophobic pocket right here. All right, let me show you the movie then. So here is the structure. So here's phenylalanine, and it sits around a lot of aromatics, so hydrophobic side chains. You would imagine phenylalanine want to be in the hydrophobic side. But there's also an arginine here below the phenylalanine, which will come back to you a little bit later. So essentially, you can imagine that if you delete this phenylalanine, you lost this contact. As it happens, the next residue 509, because when you delete this phenylalanine, 509 will take its place, right? 509 is a glycine. So it doesn't have any side chain. So there's no way to compensate for this interaction. So then you basically now messed up the hydrophobic core, this region, and that's why the system becomes destabilized. So that's the idea. So you could imagine that treating something like this is going to be very hard because of the protein folding problem. How do you find a molecule that can help a protein fold, stabilize the normal conformation of protein? So that was actually maybe even 10 years ago now, was a complete surprise that Vertex in Cambridge, Massachusetts, the company, so they found small molecules that can stabilize this mutant form. So since then, many new compounds have been developed as they are classified as potentiators. So the CFTR even get to the membrane have reduced activity, so the potentiators can restore more of the wild type activity. And then since then, correctors have been found as well, and they help the transport, the trafficking of the CFTR onto the cell surface. So you increase the activity and you increase the abundance. And so nowadays, the most popular treatment is something called Trikafta, which contains 3 molecules, so combination therapy. So 2 correctors and 1 potentiator. So these are all called caftors. That's this class of compound. They all call something caftor. Okay, so, and what I'll show you is a recent structure of mutant CFTR bound to this triple combination therapy. So this is work done by Jue Chen, Rockefeller University in New York. So they treat this, the receptor with different compounds on their own. If you treat with the lumocaftor, which is, well, this one right here, one of the type 1 correctors, it doesn't do much to the receptor. You treat the elexacaftor, type 3 corrector, it does something. You can sort of see now in this structure, one of the MBD, the one that the C-terminus is disordered. And here they are more ordered. And then if you treat with 2 lumocaftor and elexacaftor, you get better. If you treat with trikaftor, I'll show you, it gets even better. And that's why this triple combination is used for treating CF. And then compounds bind to different places on the surface. So each one of these circles represent the compound binding to different places on the receptor. This is the transmembrane region. So that's shown again right here. So delta 508, so that's a mutant, deletion mutant, bound to elexacaftor, single compound, and then overlaid onto the active wild type CFTR to see how different they are. So color codes, blue means very similar, red means 6.5 angstrom different. So you can see that the transmembrane region is pretty similar, but the MBDs are very, very different. So that's why this compound alone doesn't work very well. And then if you put in 2 compounds, everything looks much better, much bluer here compared to here, right? So you can see that the conformation is more similar. You're restoring the wild type conformation. And the difference now is only about 3 angstrom. And then more importantly, when you do the trikafta, all 3 compounds in here, you can see almost everything is blue now. So this 3 compounds essentially restored the wild type conformation of the receptor, and then MED also formed the correct dimer. So the 2 ATPs face each other, forming the correct dimer. But that's how the receptor or the transporter can become active. So here are the 3 drug molecules bound to the transmembrane region. Okay. So here is the phenylalanine 508 sitting between the MBD1 and the transmembrane region. And then in the mutant structure, so remember I showed you earlier, there's an arginine beneath the phenylalanine. Of course, now in the mutant structure, the phenylalanine is gone. The arginine moves up, take over the position of the phenylalanine. But that leaves a little pocket down here. There's something sitting here. They don't know what it is. So arginine went from here to here. That means whatever was here is now empty. So something bound there, maybe iron or water or whatever it is. So they're not 100% sure. But anyway, so overall, you can see the structure of the mutant restored by trikafta actually is very similar. MBD1 has about two angstrom movement between the two states, but very small movement. So essentially, the compound has successfully restored the conformation of the enzyme, of the transporter. And then the compound, as you see, bind to different regions. So these are the EM density for the compound. As I mentioned to you earlier, you need to look at them. So they do show them. I think generally very good quality. They bind to different regions of the transmembrane domain of this receptor. Okay, so I think this is the last slide. Let me just show you briefly a movie and then we're done, so you can have your lunch. So here is the receptor bound, the mutant receptor, delta 508, bound to the three compounds, the same color scheme. They also saw a cholesterol right here on the side. That's part of the membrane environment that stabilizes the enzyme. Down here is the two MBDs, green and cyan. And you can see they both bind ATP, and they are in the right conformation to form a dimer of the two MBDs, and that's required for the activity of this transporter. So everything worked nicely. So this actually is a cure for cystic fibrosis, because once you take this drug, you get rid of the symptoms. You don't treat the symptom, you treat the cause. So it's a remarkable achievement that to have some molecule that come in, stabilize this kind of situation. So it's quite remarkable in my mind, that achievement over the past 10 years or so for treating human diseases. Okay, so I think that's all I have for this part of the lecture. So I finished 10 minutes early, but I guess that's okay. So lunchtime for you next, and then after that, we'll do, I believe we'll do some PyMOL again. Are you interested in that? Yes. Okay, good. We'll do PyMOL for another hour or so, and then you can take your test. I'm not part of that, so. So I'm not sure how well I'll do on that. Anyway, okay, enjoy your lunch.